The major breakthrough here is that we've demonstrated in our recent paper in iScience that there is a relationship between brain activity as measured conventionally by tools like electroencephalography. There's a relationship between those kinds of oscillations and light emissions from the brain. We've known for some time that brains and other tissues emit light. We can go back to original work by
You're
you know,
rich?
these, yeah, Gurwitch, right? In the really, if we go way back, we could look at Gurwich's original experiments. But if we go back 30 years or so, we've been looking at light emissions from the brain in slice cultures. If you take a brain from a rodent, cut it up into sections, and it's still living, and you support it with nutrients, and you place neurotransmitters onto those slices, the cells will react to the neurotransmitters, right? If you put a little bit of glutamate on there, glutamate is an excitatory neurotransmitter. You would expect the cells to depolarize. They do that, and you can measure that electrophysiologically. But the other thing they do is they emit photon. And the question is, what are these photons? Are they just byproducts of physiological activity, or do they have signaling capacities? And in recent years, there have been some researchers who've put forward some pretty compelling evidence that there may, in fact, be an optical modality in the brain. That is, in addition to action potentials, there may be light signals that are shared between brain regions that act as a carrier of biological information. And we ask the question, can we measure it? Is it related to known brain activity as measured by something like EEG, electroencephalography? And what are the implications of it? Yeah, if we focus in on the energies here, I think that it should be clear that even a single photon can activate cells in visible range. It carries an energy equivalent of 10 to the minus 19th joules in energy. Even a single photon can activate one of these cells. And so, talking about ultra weak photons, which are many of them in the visible range, but also some in the UV and near infrared range, can they affect biological systems? The answer, based upon just an analysis of the energies involved, should be yes, of course. But the question is, how much tissue penetrance is going to be relevant to the actual signal transduction pathways? Like, does the photon have to go through anything, or is it simply moving to its neighbor cell, right? Like, if you have two cells right beside each other, photon emissions from one should readily affect the other. But how far is that photon penetrating? Can the light be guided? I mean, in recent years, we've seen some theoretical work by our colleagues out in Calgary, another Canadian breakthrough. We have evidence now of axons acting as potential light guides. So light can travel through tissues. Of course, it can do that. But the question is, can it bend and conform to the structures that are the brain rather than passing crudely through these structures, right? Can it be guided? And that would be pretty important in order for information to really have meaning in a biological context. And we know now that light can travel along these axons, and that should be something that we explore a little bit further.
So
Microtubules are certainly part of this. So are some other organelles, like mitochondria are part of the discussion as well. But it seems that at least the modeling suggests that myelin is probably pretty important. And myelin is, for those who don't know, the fatty sheath that surrounds neurons that we typically think of as an insulator that gives rise to the capacity for neurons to engage in what's called saltatory conduction, where the action potential moves from node to node, refreshing and maintaining that 120 millivolt depolarization as it moves along the axon barrel. But we think that actually the myelin could actually have optical properties that make it a great light guide. And that shouldn't be too surprising. I mean, we've seen evidence in recent years that the axon, as well as the myelin sheath, participate in a mechanical wave that accompanies the action potential. So there's a lot of biophysics happening here. In addition to the action potential, which is like in all the textbooks, everyone thinks of action potentials when they're thinking of neurons. There's actually a mechanical wave, and there probably is also a light, an optical wave that's occurring at the same time, all in parallel. Not only do we have electrical potentials, but in parallel to that, we have mechanical potentials, we have optical potentials. That increases the potential resolution of information by many orders of magnitude. Definitely. I think that there's a lot of unanswered questions here because an optical pathway or many optical pathways in the brain would be very much unlike the synaptic conception of how brains communicate, would be pretty messy. So suppose you have a light source. You can think of it as a candle. You could think of it as radiating outward. I could see some of those photons being carried by structures and guided, from A to B. But I could also see some of those photons passing through tissues. Some of them would be attenuated. So like there are certain wavelengths that are going to pass through less tissue than others. Some of these light guiding properties are going to be dependent upon wavelength as well. So there's going to be a lot of wavelength dependence. There's going to be a lot of spatial dependence, right? Whether a neuron is close to the surface of the head or deep within the brain, that's going to impact how far that photon travels and so on. I don't think we have the answers to all these questions, but I think there's many parallel sort of experimental questions that need to be asked to really figure out whether this is something that's biologically useful. We found that light that travels outside of the head actually has some relationship with neural oscillations at the level of the cortex. So let's say that at a bare minimum, the top, let's say 5 millimeters, this cortical region is related to photons in some sense. We have the evidence to show that the information structure of these two signals are related to each other. But what about deeper signals? We don't know. What's happening at the microscopic scale? What's happening at the level of axons? And how are glial cells related to this? This is a field that's now sort of blown wide open where we need people to address these questions, and we can't do it on our own. There needs to be a community that comes together with an interest in ultraweak photon emissions in neuroscience. There's a lot of hypotheses about where the photons are coming from. Most of the attention has been on metabolic activity from mitochondria, but there are others who've suggested processes at the level of the membrane, others associated with DNA, looking at things like DNA replication and how that impacts photon emissions. But the reality is, like, a photon can be emitted from any system that undergoes an energetic change. And anytime you have that energy change, you can have the emission of energy in the form of light. Sometimes that's in the form of heat, but other times it's visible wavelength. If we're looking for a single source, we're going to end up disappointed. There's probably multiple sources, but then again, we can probably rank order them in terms of which ones are contributing to the biggest signal. I should say that it may be that certain subsets of the spectrum that are being emitted from a cell are going to be preferentially related to certain kinds of organelles or certain kinds of molecules. If we look at different kinds of molecules in the body that we know are related to autofluorescence, like flavins, serotonin precursors like tryptophan. These are molecules that we know have interesting optical properties to them. And if you hit them with light, they will re-emit photons. There's a lot of photoreceptive molecules in the body, and they're ubiquitous, like these proteins, these neurotransmitters, amino acids, things like porphyrins, all sorts of molecules that are going to have these optical properties. So we have to look at all of these and really tease out where the contributions are coming from.